hypothesis should state your expected results. And here it is important not to describe or explain your results, but just state what results you expect from this experiment. And you must include independent and dependent variable in your hypothesis. For example, decreasing the water potential of solution makes potato chips soft. So no description, no explanation, just stating, including independent and dependent variables. The null hypothesis, it is something that you will use to avoid any bias, and it's commonly used in statistical tests like chi-squared and t-test. And the example of null hypothesis might be there is no significant difference between two groups that we compare in this experiment. After hypothesis, you can be asked to describe how you can test your hypothesis. To answer this question, you should refer to planning the experiment section, which will be later in this series of videos. Variables, as we mentioned in a previous slide, there are variables like independent, dependent variables that should be included into your hypothesis. For example, here hypothesis is the increasing temperature increases enzyme activity. Independent variable is something that you change according to your experimental setup. In this case, it's temperature. So you take different water bath with 10 Celsius degrees, 20 and 30. Dependent variable, it is something that depends on independent variable. So the enzyme activity inside of these test tubes depend on the temperature, your independent variable. That is why it is called dependent variable. So other conditions must be controlled in order to receive reliable results. So this is called controlled variable. You must control the concentration of enzyme, you must control volume, time, pH, and also the enzyme should be from the same stock. All of these are controlled variables. So it's important to distinguish between controlled variable and control sample. Control sample is something that we use in order to receive some reliable and unbiased results. And we compare control sample with other samples, and we make sure that it is enzyme that affects the activity that is happening in these test tubes. And for control sample, we can use, instead of enzyme, distilled water, or we can use boiled enzyme. Boiled enzyme is denatured enzyme. It means that it doesn't work. And it is important because we have to maintain same volume, same concentration, which are our controlled variables.